Hello, and welcome to the extraction and precipitation of plant DNA experiment. Before we get started, let's talk a little bit about human DNA. So the nucleus of the human cell contains 23 pairs of chromosomes. So we actually have 46 chromosomes per eukaryotic cell in our body. This is a little bit different than plant DNA. Plant DNA actually has only 14 chromosomes, but rather than 2 copies, there are actually 8 copies of each chromosome. So that's convenient in this experiment because we're extracting plant DNA, which actually has substantially more DNA than what is in human cells. Looking at this diagram here, this has representations but doesn't show the actual chemical structure for any of the actual compounds that make up the nucleotide monomer. So in this case, with the G representation, this is guanine, C is cytosine, T is thymine, and A is adenine. So these are the nitrogenous bases that make up DNA. These nitrogenous bases are hydrogen bonding with each other. Specifically, the guanine is hydrogen bonding with cytosine, and the thymine is hydrogen bonding with adenine. That's always the case when we have our double-stranded DNA. On the backbone, we actually have our nitrogenous bases attached to a sugar, specifically a deoxyribose sugar with DNA, and that sugar is then attached to a phosphate group. For our DNA, we have 2 strands that are running parallel to each other. For our double helix, we have major grooves, so this right here is a major groove, and we have a minor groove, which is part of the spacing with our double-stranded DNA. In this experiment, like I said, we're going to isolate DNA from the cell, so the first thing we need to do is break the cell wall. So to break the cell wall, that's why we're going to add in our dish soap. So for our dish soap, we have a non-polar tail and a polar head, which is going to disrupt the cell membrane of the cells within the strawberry. Once that cell membrane is disrupted, then we've essentially exposed the nucleus of the cell. Once that nucleus is exposed, we are going to make sure that we have salt in our solution so that our salt can strongly interact, specifically the sodium can strongly interact with the phosphate anion and overall make our DNA duplexes more neutral in solution. This will help them precipitate out of our aqueous solution because we're making our DNA duplex less polar. Please take some time to read these 2 paragraphs. It's going to explain what I'm saying in a little bit more detail. But once we have allowed our sodium cations to interact with the negative phosphate ions of our DNA, we've made our DNA duplexes, our chromosomes, less soluble in water. We can continue to precipitate out our aggregate double helix structures by adding our isopropanol. So with isopropanol, that is going to disrupt the interaction that water has with the phosphate groups more than just adding the salt, and our DNA will aggregate together in our isopropanol, allowing us to visually see that in solution. So in the lab, you're going to see that once we have precipitated out our DNA helixes, we can extract those with a simple wooden pick and pull it out of that solution. There's a calculation here using Coulomb's law to allow you to see the binding strength between DNA and sodium, and the stronger that binding strength between DNA and sodium, the higher chance that our DNA is actually going to precipitate out of our solution. All right, welcome to the extraction of DNA experiment. So we're going to be extracting DNA from one of these strawberries. They were frozen, now they're thawed. They don't look very good.
because they've got blueberry stains on them. We've got our plastic bag. This is how we're going to essentially physically break apart the strawberry. We've got our soap, our isopropanol, and right now we're heating our solution of water. So within your experiment, it said you were going to make 180 milliliters. We're going to do this half scale for the solution that we're making. So I've got 90 milliliters of water, and I'm going to add about 2.5 grams of sodium chloride. Hopefully you guys have done the calculations and you can show your work for how to make a slightly more accurate solution. And we're going to let all that dissolve, and then soon after we're going to add some soap to it. Alright, as we discussed, this is a half-scale experiment for essentially making our extraction solution. So now we're going to add soap. We need to add 5 milliliters of soap. So I'm actually going to add 5 milliliters of water to this test tube, see where the line is on here, and then just add that much soap to it. And again, I'm doing this half scale because we're only going to use this for one extraction. We're in lab. You guys could get by with everyone using one. Alright, so I've transferred 5 milliliters here. I'm going to mark the solvent line on this test tube so I know where that is. I'm going to dump that out now and add 5 milliliters of soap. Right to that solvent line. And pour it in here. I'm actually going to pour this in here to make sure we get it all out of this test tube. Alright, so there is our solution that is going to partially extract the DNA from the strawberry. Alright, now we're going to add our soap and salt mixture, 15 milliliters, in with our strawberry. So we've got 15 milliliters in our graduated cylinder now. We're going to pour this in the plastic bag. Seal the bag, and now we're going to smash up this strawberry relatively gently. So that soap can interact with these cells, break open those cell walls. Alright, now we're going to run it through this cheesecloth. All right, so we have isolated our soap solution. We're going to use 2-propanol or isopropanol as our second layer. First, actually, I'm going to transfer 5 milliliters of this into this test tube. So then we're going to put about 15 milliliters in here. And about 5 milliliters in here. And the reason why I did this, because it's a little bit easier to see the DNA in this test tube right here. You can see essentially the air bubbles trapped in this white DNA, but the DNA is definitely rising up above that aqueous layer and into the isopropanol. So we'll let that sit for a couple more minutes. Actually, if you look at the top here, you can see quite a bit more of that, what's mostly DNA. There's a little bit of soap residues, but that's mostly actually DNA there on top of the isopropanol. Alright, so we've got the DNA that definitely came up into the isopropanol. In this test tube, it's going to be hard to reach down there, so I'm actually going to add some water. That'll increase the volume of our aqueous layer, which is the bottom layer, and hopefully push that DNA up to the top. You can see that DNA jump up in there. It's hard to see the two different layers. It looks like our soap saltwater layer isn't mixing very well with the water, so we'll give that a second. And I will go ahead and isolate the DNA from here. So I'm just mixing this around in here just to basically show you that this is not soap bubbles, even though it looks remarkably similar. But you can essentially see how stringy it is, and those are essentially intercoiled DNA strands. So we'll put this in this bag here, push it around a little bit more. Probably rinse it a couple times and that red would go away, but you can see it essentially smashed down a little bit at this point.
All right, before we finish up, let's look at a couple other samples that have been done in the lab before. So this is the banana, and you can see the DNA is precipitated out of solution at the top area of that isopropanol. Also, we did not chill the isopropanol. We probably could have gotten a slightly higher yield because more DNA would have precipitated out of solution with chilled isopropanol instead. For the next sample, this is a crushed kiwi, so this is DNA precipitating out of that. This is still a shot of a different extraction of DNA from strawberries. Now note that in this lab I did not go back to the test tube that I had, and we'll go back to the video there. So in this video you can see that when we added that water, a lot of the DNA actually dissolved in solution. So since isopropanol is somewhat soluble in water, especially if both solutions are warm, the vast majority of DNA actually dissolved in that solution in this case. So I could still extract a little bit of DNA from the top of it, which you can see there, but it's just a tiny amount because of the solubility of DNA in water, especially when the salt water becomes diluted, and especially then when water is intermixed with the isopropanol. So again, you can still see a very small amount of DNA, but that's actually dissolving in solution pretty quickly in that test tube.